Hello, and welcome to Cognito Key. Today, we're going on a little adventure into the microscopic world. We're going to tackle a really cool question. How on earth do scientists actually see things that are, for all intents and purposes, invisible? I mean, think about it. You've got this super powerful microscope, right? But when you look through it, most of these little microbes are basically ghosts. They're tiny, they're see-through, and they just blend right into the background. There's just no contrast. So, that's the big problem, right? And the solution is, well, it's brilliant and incredibly colorful. Here's what we've got on the agenda today. We're going to start with this whole invisibility problem, then we'll get into the chemistry behind how we solve it, and finally, we'll check out the different staining tools that scientists have in their toolkit. All right, first things first, let's talk about what staining actually is. This is the absolute core technique that makes the microbial world visible to us. You can think of it like giving these microbes a dye job. Seriously, we're just adding color to create contrast. And when we do that, these tiny little guys and all their different parts just pop. It makes them stand out so we can actually study them properly. Now, it's not as simple as just splashing some dye on there. It's actually a pretty neat three-step process. First, you prepare what's called a smear. That's just spreading a thin layer of the microbes on a glass slide. Next, you fix them, usually with a bit of heat, which basically glues them to the slide so they don't wash away. And then finally comes the fun part, applying the stain. Okay, so the big question is, how does this actually work? Like at a chemical level, how do these stains not only add color, but also stick to the cells? Well, to figure that out, we need to dip our toes into a little bit of clever chemistry. Believe it or not, it all starts with something called a benzene ring. And as you can see, it's completely colorless. So by itself, it's not much help to us. To get that color, scientists add a special chemical group called a chromophore. Think of the chromophore as the pigment. It's the part that gives the molecule its color. Once you add it to benzene, you get what's called a chromogen. So now we have color, but we're not done yet. See, just having color isn't enough. If you just pour it on, it'll wash right off. The chromophore needs a way to stick to the cell. And that's where the third piece of the puzzle comes in, the auxochrome. This little guy gives the molecule an electrical charge, kind of like a magnet. This charge is what lets it bind to parts of the cell. And just like that, you've got a working stain. Okay, so we know how to make a stain. Now let's look at the simplest way to use one. This is called, well, simple staining. And it's perfect for when you just want a quick look at the basic shape, size, and how the bacteria are arranged. What's cool is that even though you're only using one dye, you can do it in two different ways to get opposite results. First, there's direct staining. Here, you use a stain with a positive charge. Since bacteria cells are negatively charged, opposites attract. The dye sticks right to the cell and colors it in. Then you have indirect staining. For this, you use a negatively charged stain. And what happens when you put two negative charges together? They repel. So the stain gets pushed away from the cell and colors the background instead, leaving the bacteria clear and easy to see. But simple staining has its limits. What if you have a mix of different bacteria in your sample and you need to tell them apart? Yeah, simple staining isn't going to cut it. For that, you need to level up to something called differential staining. And as the name suggests, it helps you, well, differentiate. And the absolute king of differential stains is the gram stain. It's the cornerstone of microbiology. Seriously, it divides almost all bacteria into two big groups, gram-positive and gram-negative. Here's how it works. First, every cell gets stained purple with something called crystal violet. Then you wash it with alcohol. Now this is the key step. Gram-positive bacteria have these really thick cell walls that trap the purple dye, so they stay purple. But gram-negative bacteria have much thinner walls, and the alcohol just washes the purple right out. Finally, you add a pink counterstain called safranin. The gram-positive cells are already purple, so nothing changes for them, but the now colorless gram-negative cells soak up the pink. So in the end, you get purple guys and pink guys in the same view. Another really important one is the acid-fast stain. This is for a special group of bacteria like the one that causes tuberculosis, Mycobacterium. These guys have this waxy layer in their cell walls that the gram stain just can't get through. So with this method, you use a strong red dye and heat to force it past that waxy layer. Then you try to decolorize it with acid alcohol. But that waxy layer that was so hard to get into, it's also great at trapping the dye inside. So the acid-fast bacteria stay bright red. Everything else loses its color and then gets counterstained blue.
Okay, so we can classify whole bacteria. But what if we want to zoom in even further? What if we want to see specific little parts of a single bacterium? Well, for that, we have what are called special stains.
These special stains let us see all the cool gear bacteria have. We're talking about things that help them survive, move around, and even cause disease. For example, there's the capsule, which is kind of like a slimy shield. Then there's the endospore, an incredibly tough survival pod. And you can't forget the flagella, which are like tiny little propellers they use to swim. So, to see that slimy shield, the capsule, scientists use a negative staining technique. It's really clever. The background gets colored, and the cell itself gets colored, but the capsule in between doesn't take up any dye. So what you see is this beautiful, clear halo around the bacterium. And seeing that halo is super important, because it's often linked to how dangerous a bacterium is. And what about those super tough endospores? To see them, you have to use heat to literally force a green stain into their tough outer coat. After that, the rest of the cell is washed out and stained pink. The final picture is really striking. You get this bright green spore sitting safely inside a pink cell. It's unmistakable. Okay, so we've gone through simple stains, differential stains, special stains, a whole lot of techniques. But let's take a step back. Why does any of this actually matter in the real world for science and for medicine? Well, it's definitely not just about making pretty pictures for a lab notebook. Staining is a critical tool. For a doctor, it can mean quickly figuring out what kind of infection a patient has and choosing the right antibiotic to fight it. For scientists, it's how they tell different species apart. And for researchers, it's how they study the very things, like capsules and spores, that make these microbes so successful and sometimes so dangerous. So you see, these simple, colorful techniques are what transform a regular microscope into a powerful gateway of discovery. They allow us to see, to classify, and to truly understand the huge, invisible world of life that's all around us and even inside us. Thank you so much for exploring the science of staining with me today here on Cognitu Key. If you enjoyed this journey into the microscopic world, please give this explainer a thumbs up, share it with a friend, and don't forget to subscribe for more deep dives into the amazing world of science.